Our next speaker is another boy who fulfilled, as I said, his Jewish mother's dream. This is Dr. Gad Asher. Gad received his MD degree from Tel Aviv University Sackler School of Medicine in 1998. During medical school, he spent 3 summer internships at the Weizmann Institute, followed by a residency at the Tel Aviv Sourasky Medical Center. Like Eran, he also joined our PhD for MD, and in 2006, he received his PhD in molecular genetics from the Weizmann Institute. After a postdoctoral fellowship at the University of Geneva, Switzerland, he joined the Weizmann Institute a year earlier than Eran in 2011. And Gad is studying something that I'm sure bothers everybody who flew in and have a hard time getting his time regulated. We are all jet lagged. And the title of his talk would be, The Time. Who's got the time? Please, Gad.
So, good morning, everyone. It's a true pleasure to attend this event, and in fact, to see in person and meet all of you. It's also an honor to have Dr. Young here in the audience, because he's also a true leader in the field of circadian rhythm, so he might correct me in my talk. So my lab is working on biological clocks, specifically on circadian clock, and I will walk you through this topic during my talk. So have you ever asked yourself, why do you wake up in the morning? Why do you go to sleep at night? In fact, why do you perform different tasks differently during different times of the day? So in fact, our body has its own timetable. Now it's 10 in the morning, so it's the best time to give a seminar because our ability to concentrate it's at its peak. So thank you, Israel, for giving this slot to me. If you need to go to the dentist, I would strongly advise you to fix an appointment at around 2 in the afternoon, because at around 2 in the afternoon, your body is least sensitive to pain. Later on, there is increase in blood pressure, body temperature. Towards the night, most of us go to sleep. Around 4 in the morning, we have the highest risk to have an heart attack because there's increase in blood coagulation, but also the highest risk of baby's delivery. So these changes throughout the day are actually the outcome of the function of different organs in our body. So there are daily changes in the function of different organs, and this consists of the cardiovascular, renal, body temperature, endocrine system, and many other organs. These changes are not a simple response to changes in light and dark cycles that normally occur in our day. These changes are governed by an internal clock. In fact, all light-sensitive organisms, from the bacteria that Eran mentioned, through plants, flies, mice, and humans, all of them have these clocks. And to convince you this case, that this is indeed the case, I will show you the classic experiment. This experiment was done on all of these organisms, including humans. So mice are nocturnal animals. Unlike us, they are mostly active during the dark, during the night phase. It's very difficult to catch a mouse in the night. By contrast, they sleep during the day. So if you take a mouse, we can very easily monitor the activity of the mouse. We can put it in the cage and monitor the activity. And I will walk you through this slide. So this is, each line represents a day. The blue represents the dark phase, the white, the light phase, and the bars indicates the activity. So you see, if you expose the mouse to a 12-hour light-dark cycles, the mouse is mostly active during the night, as expected, and is sleeping during the light phase. But what will happen now if we turn off completely the light? The mouse is devoid from any external timing cue. The food is freely provided. Nobody knocks on the door and indicates the time. So if we turn off completely the light, you can notice that these 12-hour rest activity cycles remain. So at the first day, the mouse starts to be active at 8 in the evening, like before. Later on, it starts to be active a bit earlier and goes on and goes on. So the activity of the mouse under this condition is not random. Something tells him when to start to be active. But you notice that it's not exactly straight line. And this indicates that this clock or this time machine does not measure exactly 24 hours. In mice, it's measured 23.7. In fact, in human, it measures more than 24 hours, 24 hours 0.8. So this is why it's called circadian time, or circa in Latin it's about, and diem is a day. So it's clear that we are living in a world of 24 hours. So such a clock is useful, but it needs to be adjusted in time to time with the geophysical dial. And this is done by exposure to light. So first of all, where is this clock located? The clock located in the brain, in the region which is called the suprachiasmatic nucleus in the brain, and the neurons, the same neurons that we have talked about yesterday, contains oscillators which cycles with a frequency of about 24 hours. There is also the input which comes through the eyes that resets these oscillators every day and synchronizes them. So I've been talking about clocks, and you are all probably wondering, what are the digits? What are the dials in this clock? So I will tell you a short anecdote from the lab where I did my postdoc in Geneva, the lab of Dr. Uli Schibler, that will illustrate this. In fact, we scientists, we believe that we can design the perfect experiments. But in many cases in science, serendipity can lead to major discoveries. So about 30 years ago, the lab of Dr. Schibler was working on a complete different topic. They were working on transcription. I will not go into the details. And Dr. Schibler was interested in purifying a protein from mouse liver, which is called, the protein is called DBP. And to this end, he recruited a young PhD student. This young PhD student was coming from a farmer family. He was used to wake up early in the morning, working in the farm, and likewise, he was coming to the lab in 6 in the morning, sacrificing the mice and purifying the protein. After 4 years of hard work, he managed to do so, and this was indeed a major achievement. It was published in a very prestigious journal, and the scientific community was very excited about it. So Dr. Schibler decided to continue to work on that. The PhD student completed his studies and left the lab, so he recruited an American postdoc to continue to work on that. So the American postdoc joined the lab, and the first thing he did, he tried to reproduce the data that the former student did. So he also worked very hard. He worked about 6 months, and he didn't manage to produce it. He didn't manage to purify the protein. This obviously caused a major embarrassment in the lab, and they were about to announce and inform the scientific community that actually the results are probably not true. But then Dr. Schibler noticed that this American guy, he had a complete different lifestyle from the former student. He really liked the nightlife of Geneva. He used to go out drinking late in the night, sleep late in the morning, and come to the lab around 2 in the afternoon. He would start the experiment in 6 in the evening. So they decided to repeat the experiment and do it the same, but now early in the morning. And to their surprise, they managed to reproduce the data and produce the protein from the liver. So just to show you, so this is the original plot. This is the intensity of the protein. The band indicates the intensity of the protein. So indeed, in the liver at 8 in the morning, there is really high level of the protein. But if you look at 8 in the evening, you can barely detect it. So the digit, the dials, are the protein which oscillate or accumulate in a daily manner. So nowadays, we know that up to 15% of our proteins are accumulating in a daily manner. Some of them accumulate in the morning, some in the afternoon, some in the evening. So you can imagine that if you take a same cell and look at it in the morning and in the evening, you will see a complete different picture. So the second revolution in the field occurred about a decade ago, when it was discovered that there is not only one clock in our body, there are millions of clocks in our body. Every organ in our body has a clock. Every single cell has a clock. Moreover, we can take cells from each one of you here in the audience. We can keep them in culture, in a petri dish, and we can follow the function of the clock. So I can actually not only tell the time of your clocks, and also I can also tell if your clocks are functioning properly, and if indeed they measure 24 hours and not 26 or 22 hours. So this is the original classic experiment. So again, the intensity of the band indicates the level of the protein, and you see that period 1 protein accumulate in a daily manner. It peaks every 24 hours. So nowadays, we and others developed more sophisticated and elegant way to follow the function of the clock in living cells, in single cells. So we created by molecular genetics tools that enable us to follow the function of the clocks in living cells. So we use a fluorescent protein, and we follow the accumulation of this fluorescent protein in the cells for several consecutive days. And as shown here in this movie, so the time is indicated in here, and these are the cells, the fluorescent protein, and just follow the accumulation of the fluorescent protein. So now it goes down. After 24 hours, all the cells are green, and then it's diminished again, and it will go up again. And this is a very powerful tool. But like this, we can actually tell each one of you how is the clock functioning, and if you indeed have a good clocks. So the current idea is that we have a master clock in the brain, which receives input through the eyes and is sensitive to the light-dark cycles. This master clock synchronizes clocks which are resides in peripheral organs, and these organs in turn regulates our physiology and behavior. So how do we kind of build a clock? You know, you need to be really a genius engineer to engineer or build a clock which will still measure time accurately and also responds to external timing cue. So the basic principle how this clock functions is a basic principle which is called a negative feedback loop. This principle is quite prevalent in biology, in chemistry, and also in engineering. For example, the thermostat that you use in your air condition is working the same. You know, if you set the temperature to a certain temperature, the air conditioning is cooling the room. Once it's reached the temperature, it stops cooling. Then the room starts to warm up, and then the air conditioner starts to work again and cool the room again. So in biology, obviously, it's slightly more complicated. We have an activator, which is drive the accumulation of a protein which is called repressor, and this repressor inhibits its own accumulation. And it looks like this. So the activator induces the accumulation of the repressor. The repressor is accumulating. Once it's reached a certain threshold, it binds the activator and inhibits activity. So the repressor is slightly degraded and goes on, and then the activator is released to induce a cycle. A new cycle. And like this, we generate another and another cycles. Like this, we generate cycles in protein accumulation. Of course, in biology, it's much more complicated, and we have several feedback loops which are interlocked in each other, and this feedback loop generates cycles of about 24 hours. So why do we need clocks? You know, clocks are conserved in evolution from bacteria to human. So it's clearly not a caprice of nature to keep this clock in humans also. So we have evidence from several sources that these clocks are indeed important. So the evidence are the pathologies that we observe when these clocks are non-functional. So this evidence comes first from shift workers, for example. These people are working actually in the time they are not supposed to work against the internal timing system. The other source of information comes from animal models in which they have disrupted clocks. From both these sources, both the genetics and the environmental sources, we know that these clocks are not only important for sleep, when disruption of rhythmicity results in sleep disturbances, but also these clocks are very important for maintenance and prevention of malignancies, metabolic disease, and accelerated aging. And this is actually the big revolution in the field, because for many years it was not really clear why do we need this clock, and nowadays we know that they are really important, and I will focus later on metabolic diseases. Another thing that I would like to mention is that I mentioned that there is a master clock in the brain, and also there are numerous clocks in peripheral organs. So obviously this condition is harmful for the organism, because the clocks should be synchronized. They should work in unison, in harmony. So how does the master clock in the brain synchronize all these clocks that they will all work together? So this is mediated via several redundant pathways, but there is one main principle that kind of couples all of these clocks. So mice are animals that are mostly active during the night, and obviously they eat about 82% of their food consumption during the night. If we look at the clock in the master brain and the clock in the periphery, both shows exactly the same time. But what will happen now if we will force the animal to eat exactly during the opposite time? We will provide the animal the food exclusively during the light phase. So for the master clock in the brain, it does not matter, because for the master clock in the brain, the main timing cue is the light-dark cycles. But what about the clocks in the periphery, in the liver, for example? These clocks are completely inverted. They show exactly the opposite time. So this indicates that for the clocks which reside in peripheral organs, such as the liver, the kidney, the spleen, these clocks are mostly responsive to feeding. So this brings us to the idea that the master clock tells us when to eat, and then feeding time actually resets the clocks in the peripheral organs. So besides its scientific merits, it also has some practical implication. And as Israel said, jet lag is a major problem. So how can we use this knowledge to fight jet lag? So first of all, what is a jet lag? Jet lag is the inconvenience that is the result of the fact that our internal clocks are not in accordance with the geophysical time. Once we fly from Tel Aviv to New York, our body clock still thinks that we are in Tel Aviv, but the outside shows a different time. So it takes few days to the internal clock to be resetted and to be adjusted to the new time zone. So can we speed this process? Yes, indeed, we can do so. We can do so if we will fast during the flight, and we would eat according to the time of the destination. So if we arrive to New York in the morning, we should have the breakfast in the morning, the lunch in the lunchtime, and the dinner in the evening. And like this, we will be able to reset this clock much faster and decrease the inconvenience which is associated with jet lag. So during the last part of my talk, I would like to dedicate it to the research I've been conducted in the lab during the last year and a half, 2 years. So we were really interested in the connection between circadian clock and metabolism, and in particular in lipid homeostasis, namely the accumulation of different lipids in the liver. So we did a huge effort to quantify hundreds of lipids in the liver, and indeed we found that there are lipids that accumulate in a daily manner in the liver. So one family which was really striking is the triglyceride. You are probably familiar with them, and they accumulate in a daily manner. And at the same time, we also monitored the feeding habits of the animals. So as I mentioned before, mice mostly eat the food during the dark phase and only about 20% during the light phase. So we tried to fiddle around with this system, and we looked what will happen if we now limit the food accessibility only during the dark phase. So you see the mice eat only during the dark phase, and we can change the accumulation of the triglyceride levels. But what was really striking in this experiment, that though we changed the feeding time of the animals, on the one hand, the one group was eating exclusively during the night, while the other was freely eating, they both consumed the same amount of food. But when we quantify the triglyceride level, within a week, we saw a 50% decrease in the amount of triglyceride in the liver. So as Eran mentioned, fatty liver is the accumulation of triglyceride in the liver. It's a very common disease which results in malfunction of the liver. It's highly associated with a lot of morbidity and mortality, obesity, and diabetes. There is no available drugs that can do this thing, decrease the level of triglyceride in 50% within such a short time without the side effect which is associated with many of these medications. So by merely changing the time of the feeding, we can actually modulate the accumulation of triglycerides in the liver and probably many other metabolites. So in line of that, recent studies have shown that if you take mice and feed them high-fat diet, obviously they will gain weight and will develop this metabolic syndrome which is associated with diabetes, insulin resistance, and all the other effects. If you take the same animals and give them the same high-fat diet, but you now limit it only to the dark phase, they can only eat during the night, the mice will not gain weight. They will remain lean. So it's only the time that changes. So most of us are living in this notion that, it's important what we eat. And the take-home message that I would like to give you today, it's not only important what we eat, it's also very important when we eat. So obviously, we are far from doing it in humans, but if we extrapolate this result from humans, it means that we should eat during the active phase. And we should keep these feeding fasting cycles. In the past, food was not available like today, and we were actually practicing these feeding fasting cycles. But nowadays, we can actually open the fridge in the middle of the night and take whatever. So the next time you open the fridge in the middle of the night, please think about it twice. I would like to introduce you with another concept. So the last 2 days, we talked a lot about the personal medicine. I would like to introduce you to a concept which is now emerging, which is called time-dependent therapy, or in professional manner, chronotherapy. So as I mentioned before, many proteins accumulate in a daily manner. Many of these proteins participate in the processing of many of the medication that each one of you takes. So if you think about it, it is conceivable that if you take a certain medication in the morning or in the evening, it will be processed completely different. And indeed, animal studies and even clinical studies have showed, at least for chemotherapy, that if you take the same chemotherapy in the morning and in the evening, the outcome will be completely different. You can get much better outcome if you take it at a specific time during the day, and also you can significantly reduce the side effect. So this is a field that is now emerging, and we and others are trying to address it by collecting a lot of data about the changes in the protein accumulation, in the metabolites, and we actually can predict and test these things in the lab because we can test it on the animals and hopefully in the future also in humans. So I would like to end my talk with the acknowledgement. I would like to thank first the 2 stars of my talk, which are my lab members, a group of highly motivated and talented students. I would like to thank the Weizmann Institute, who provided me with all the means to establish a state-of-the-art lab. And I also would like to thank you for your contribution and attention. Thank you all.
So we've got the time for a few questions, please. Yes, at the back.
Can you hear me? When I set my alarm in the evening, say for 7 o'clock in the morning, and I know I have to get up and do something, I will wake up anywhere from 5 to 15 minutes before the alarm goes off, almost consistently. If I don't set the alarm and I don't have anything to do that morning, I can sleep all the way to like 9 o'clock.
So, you
Why?
you imply that your clocks are not working well. So I will admit that I don't have a good answer besides saying, okay, maybe I can take a few cells from your body and I can take him to the lab in Israel and call you in a few days and tell you, okay, your frequency of the clock is not so good. I will give you an example. If it's a really nice anecdote of one of my colleagues. You know, there's a lot of connection between sleep disturbances, waking sleep time, and circadian clock. And one of the ways to address it, you know, with humans, it's actually to try to see and collect samples from humans who have sleep disturbances and see whether indeed what I presented to you is true and whether they have a non-functional or dysfunctional clock. And one way to do it, and it's really nice, is just to try to recruit these people, and it's not so trivial. So one idea is to put an ad in the TV in 2 in the morning. Actually, it's very cheap to put an ad in 2 in the morning because no one wants to advertise something in 2 in the morning. And in this ad, my colleague wrote, please call me if you see this ad. And like this, he collected a lot of samples from people who have sleep disturbance. He took the cells, used the methodology that we and others developed and I showed you, and he measured the period of this clock. And he found a very strong correlation with long and short period with sleep disturbances and with this problem of waking up or going to sleep. So this is my best answer.
One last question. Yes, please.
I have the microphone. Hi, can I ask you a question? Those of us who have teenagers have watched in astonishment how they go to sleep at 2 a.m. and get up at 2 p.m. What can we do to change that clock?
It's always a tough question that's coming from a general audience. I don't have a good answer for that. I just advise you to force them to go to sleep early. But, you know, I would take it to another direction. You know, our clocks are, in general, very robust. But you all know that as you get older, you know, the clocks are functioning less well. And this is evident by the fact that the elder you are, you sleep less, and you wake up early in the morning. So when you are really young, the clocks are not working well, and when you are very old, the clocks are not working well. When you are a teenager, you are a teenager.